Hello and welcome to the NGS Webinar Series hosted by the Next Generation Sequencing Competence Network. Along the series, you will be hearing from scientists and clinicians from various fields of research. However, they are all connected by sequencing. Who would have guessed? We bring you to journeys where researchers are telling us about their sequencing data harvesting and bioinformatics analysis and surely results and perspectives. How do they do it? What is it good for? Watch and let us know. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We'll leave the link in the footnotes, and here is QR code for your fast access. Welcome aboard. Find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
All right, so to get started, our goal was to have rare disease diagnostics with Nanopore. And for that, you always have to set up your whole laboratory and bioinformatics setting to accommodate this new technology. So the clinical part might be quite similar, except that you need high molecular weight DNA. Sequencing is obviously new. You have very new data with different quality, and well, there's a lot of optimization, but I will not talk about that part because I'm a bioinformatician, so I will talk more about the right part. What we had to update were the whole pipeline from storage, which at least the raw data is like 10 times larger, two different settings. You should add here base calling and so on, alignments, the variant prediction, and then also all the annotation of the variants all have to be updated, because as we later see, we will get much more structure variants. We have no reference data for nanopore structure variants. So functional annotation becomes much, well, harder for now until we have large cohorts to actually have a background. Clinical interpretation gets more complicated because we are now looking in regions we haven't been able to look into. So all of that has to be updated. We have a clinical decision support system here that we develop in Tübingen that is connected. It's called GSWAR, connected to a pipeline called MaxSAP, and we have updated all of them to include Nanopore now. So to show you a bit how that process has been going and how we developed all the different ideas, let's first look at what is different for Nanopore compared to the old school short read we have been doing for 15 years. So first, we have now much longer reads, 20 kb, 25, if you want, even 100 kb. So we need new alignment strategies, or we can alternatively go for genome de novo assembly and then whole genome alignment. So it's a completely new possibility. Whatever part you use, Minimap or some assembler, Fly or whatever, you can now do haplotype phasing, which was near impossible with only short reads. Here with the long reads, it's much easier, and we will have much more variants, as I mentioned, and for free we get CpG methylation. So in the same run for the same base calling, if you use the 5-base or 6-base model for Dorado, you get methylation. And you can also phase the methylation data, not just the variant data, and can then have frequencies of methylated cytosines on each site. Okay, so lots of new things. If we look at the haplotype phasing, we now get a very different picture. If we actually haplotype phase the reads, we can look, this is a screenshot from IGV, we can now look at haplotypes instead of reads, which makes it much nicer to actually look at families and larger families, because you don't have all these tracks with hundreds of reads, but you just get one bar for each allele. And then you see inheritance very easily, like this insertion from the mother, same in the father, both heterozygous, is inherited by both daughters. This is not a causal one. I just chose that example because it nicely shows how to use this to very quickly get an idea of a specific region and the haplotypes that the children's inherited. So in my opinion, that's quite a change in the way we look at genomes. To do that, we had to develop pipelines, as always in bioinformatics, and we had to test a lot of tools, finding out which ones are good, which ones not. For that, our NGS Competence Center, the NCCT in Tübingen, specifically Kaspar, Jakob, and Vladislav, made Nanopore pipeline and tested it. So the initial pipeline was done by Kaspar. And then together with Elena, who developed all the wet lab parts, they looked into which ones are the best tools, for instance, for structure variants and so on. Once all these tools were tested, then Leon and Mark translated it into our standard pipeline, MaxSap. So the testing pipeline is called MacLR. Both can be downloaded on GitHub. And in MaxSap, we then only have the tools that really worked well, and everything else was kicked out. Finally, we have to adjust in our clinical decision support system where you can look at the variants, filter them, and so on. We had to include long read filtering and long read analysis, and especially also methylation analysis, and we have done that by now. It's fully supported, and you wouldn't actually realize the difference if you just look at small variants. But if you look at structure variants, repeat expansions, or methylation, it is different. So in our system, we had to update a lot of stuff. HPO terms not, they actually stayed the same, but the CNV calling and the SV calling and the repeat expansions and structure variants, all of them, we had to implement new algorithms in MaxSap and also for RNA, but that's not a topic today. Finally, we obviously generate reports out of the system, and that will have to be updated as well, especially once we get better, like repeat expansions or duplicate genes and so on. We will slightly adjust the clinical reports, but mostly they are the same. You report one causal variant using the system. So to summarize, we had to change quite a lot. If you want to accredit Nanopore as a laboratory developed test for diagnostic testing, you get a visit from the DAX, and then you have to basically show that all the different parts of the test, from sample collection to robotics to sequencing, data storage, analysis by computational analysis, and then analysis by the geneticists, is ready to actually used in diagnostics. So it's robust. If you repeat a test, you get the same result, and so on. And we needed new databases for long read data filtering. That has been done, and Tobias Haag's group accredited it in, I think, November last year for diagnostics. For us bioinformaticians, the most important contribution is quality control and benchmarking, and I will discuss that specifically in this talk. All right, so first of all, to exactly do that, not just in Tübingen, but in many German university clinics, several clinics that you can see here founded the so-called LONGER consortium. So for Long Read Germany, or Clinical Long Read Genome Initiative of Germany. So it's for now a German initiative to evaluate clinical application of nanopore sequencing. We started with 4 university medical centers that get support from ONT directly. So we get flow cells to make 1,000, in total 1,000 diagnostic nanopore cases. And we recently got a lot of interest from others. So in that graphics, we already have Mainz in here, but we also got interesting parties from Munich and other universities. So that picture will hopefully grow in the future. although the direct support from ONT will probably not be expanded. So free flow cells will only be there for the first samples. We rely on a clinically well-characterized cohort of rare disease patients. Initially, we did 80 samples for benchmarking, including solved cases to see how good we are, and including the typical GIAP, genome in a bottle benchmark samples, HG001 and 002. But in the future, we want then to do unsolved cases. We want to demonstrate the diagnostic value. That's the most important thing. Why switch to long reads if there is none? Sustainability is also important. Prices and throughput from the sequencers. Can you actually sustain it if you have several thousand people per year, several thousand patients? We need to accredit and benchmark. I just mentioned that. And have SOPs for both wet lab and bioinformatics, and also for interpretation. So there's a lot of stuff to do. Quickly introducing the pilot study we did with 80 samples. I mentioned 2 genome in a bottle, GIAP cell lines. The NA12878, everyone knows. HG001 is the other name for that one. And we included also the second big GIAP, HG002. Because I think it has better annotation for structural variants, if I remember correctly. Then we included 68 patient samples where we knew the outcome, and we used the latest R10V14 chemistry. The clinical cases we included were especially repeat expansions, low complexity regions, genes with a pseudogene copy, like a paralog, where you have repeat problems in the alignment, pathogenic haplotypes, the obscene cluster for eye disease, and imprinting disorders where we can utilize the methylation information. So all hard cases, we didn't include actually the super easy stuff. So here's an overview of the ones from Tübingen, which I quickly included, mainly to give you some idea of the quality and depth we try to achieve. So normally we try to get 15 to 20 kb. Here are some shorter ones, mainly because we used already extracted DNA for Illumina, where people did not take care of having the highest molecular weight. So overall, our test cohort was a bit shorter fragment size than we normally would like. This would be optimal, 20 kb. The yield, you can see there's a big range from one flow cell, but we can actually reach 160 GB. And well, there were some not so great flow cells, but I will show more about that. So you get an average depth, and you can see what fraction of the genome was covered with at least 20x. And you can see that if you reach 35x coverage, most of the genome is covered at least 20x. But there are these problems where you have less than 30x, then you have big gaps in your genome. So very important, go beyond 30x, try 35 or even 40. There will be more reasons for that I will show later. And then the overall runtime of the analysis, that has changed. We just recently got a new super NVIDIA computer, which probably halves that, the runtime. All right, so why is the yield of flow cells sometimes great and sometimes not so great? As we said, 40x is optimal, and for that you would need about 140 GB output or 130. The absolute minimum, I think, is 30x, for which you need 100 GB, considering that some reads will have to be removed for quality reasons. And then down here at the 80 GB level, that's not what we like. And what we see is it's mainly a flow cell issue. Some flow cells have this nice behavior. Here's the top up. And some flow cells have this bad behavior that they rapidly drop, also not linearly, but like this quicker drop at the beginning where the flow cell, the pores clog or something. We don't know. I guess you cannot do much about it except for even doing more top ups, but even then you will never get the output of a good flow cell. So I cannot give you a good hint. except for maybe sometimes just replace the flow cell. Very important, you have to do this top up. So after one day or after each day, if you top up, you get much more output if you flush the flow cells and top up with new DNA. If you have questions for that, please, you can ask Elena from our institute, or also Florian Kraft from Aachen has very good experience with that. Now, benchmarking, clearly we want to know if we are at least as good as with Illumina or even better, and we use Genome in a Bottle. They provide these very deeply sequenced reference cell lines, and then they have multiple samples. Most famous one is probably this one here. And then they also provide others from different ethical backgrounds. There's a high confidence regions, about 70% of the genome, which with short reads is fairly accessible. And there are the not so accessible regions, which are now very interesting for long reads and also for structure variant calling. Unfortunately, for our latest genome version, GRCH38, I think there's still no official release of the structure variant benchmark set, but there's an unofficial release if you're interested. Good. So why is it good? Because people sequence these cell lines with every sequencing technology feasible, including even non-sequencing stuff like BioNano and also PacBio and BGI and Illumina and single cell and so on. So these have been really well assembled and annotated so that the variants you get there, you can consider gold standard. So every benchmark is based on the assumption that what GIAP delivers is correct, which might be in 0.001% not the case, but mostly it is. So looking at the SNV and indel precision, I have to say my plots, I didn't renew them. They are still with Guppy, but I will show later that Dorado, the latest base caller, doesn't make such a big difference. So for SNVs and indels, the precision first, how correct are our calls? You can see that for SNPs, it doesn't matter which algorithm you use, CLAIRE3 or deep variant, about at 30 to more like 35x, you reach close to 100%, and at 40x, you are basically 100% in both benchmark sets, both cell lines. For indels, they look very different. You are still improving even at 60x, and you never reach genome-wide precision that is higher than 95%. So that is definitely a problem, as you might all know. And it also makes a big difference which algorithm you use. Again, a disclaimer, this is not the latest deep variant 1.5, but the version before, which was still called Pepper Margin deep variant. We are right now testing the latest, but maybe at the ESHG, I can present these new informations. Looking at the recall, you can see same picture. In the SNVs, doesn't matter which algorithm, at around 30 to 40x, you get 100% recall. So that's amazing, but indels, this time it's the other way around. Precision, CLAIRE3 was better. Recall, deep variant is better. But again, on a genome-wide level, this is not very satisfying, unfortunately. So on the new Dorado base caller, I just got the latest update from Leon Schütz here, who analyzed this data together with Jakob Admart from the core facility. Again, same reference. And you can see that this new model, so we switched to Dorado and we use now the 5-base instead of the 4-base model for methylation. And we gained 1% on recall and 1% on precision. For precision, that's not bad. It brings us closer to what we have with Illumina. But for recall, that still doesn't cut it. And same for protein coding, the improvement is even smaller. So I would not say that this is a game changer. It's getting better, and that's good. Nanopore consistently gets better every year, but it's not a game changer yet. Good, so duplex reads. ONT presented them already a while ago, and that is just taken from their website. Here is the idea. You basically sequence the reverse strand immediately after the first strand, but other than in PacBio, there's no like connection. Like no, PacBio is totally different. They do a circular consensus of the same strand over and over. But I don't know why it cannot be directly connected. It is still a stochastic process that the reverse read enters the pore. It's not forced in, but it's more likely, I think, because they are closer to the pore than other fragments. That's how far I understand that whole process. You can probably read that in more detail in the publication. Anyway, the duplex reads, if you can use them, you can correct the forward and the reverse strand, and you get a much cleaner output than with the simplex reads, or much, much better than with the old R9 V4 chemistry. So that looks promising. Is it real? So Caspar made some analysis and Leon to see if that is the case. The first thing we realized is that if you look along the cycles for very long reads or very long, medium long reads, you can see that similar to Illumina, there is some drop in quality over the reads. That's interesting. I wasn't aware before that even Nanopore has a reduction in quality along the read length. But if you do duplex, because you now sequence from both forward and reverse strand, this is gone, this problem. So you get good quality from start to end. And that obviously also increases the Q30 automatically, just having that. Now there is the update of the error rates for insertions and deletions. You can see here, we get a 75% less errors. Or here even more, 80% less deletions, about 75% less insertions. So that's very good. ONT even reported a 10-fold reduction. We see a 5-fold. So, well, we have to see if we can improve the algorithm a bit. It also improves SNFs, here about 3 to 4-fold. But the most important for us is that we don't need soft clipping anymore, because of the previous slide you have seen that the reads are now good to the very end. So with the simplex reads, almost 100% of the reads are soft clipped in the end, so they are not aligned to the end by Minimap. With the duplex reads, now only 12% are soft clipped, meaning almost 90% of the reads align in full length. And this will be very nice for also the RNA-seq and cDNA sequencing and isoform detection. So good results. Why is it not the super solution yet? It's because we only have, in our case, less than 20% duplex reads. And, or I think even in most of our cases was only 10%. Again, in Aachen, they had a bit more luck. I think they reached Florian's lab, they reached like 30%, and Nanopore's promising, ONT is promising up to 50% in future improvements of the kit. And then indeed, this would be the solution to solve most of our problems. Finally, maybe you recognize this guy here, that's German Demidov. He found this on the weekend, a tweet by a group that claims they have now the perfect error, haplotype error correction, which brings us from 20 to almost 40 Q. If that is true, there is a GitHub. Here's the description: highly accurate haplotype aware deep learning tool for error correction for R10 flow cell. They prefer reads above 10 KB. If that is true, and German is currently testing that, well, maybe it solves a lot of our problems, but there is no paper, so we cannot tell you if this is real or just claiming a lot. And well, we might know in a month. Okay, coming to some examples, we have structure variants. Here you can see with Nanopore, they look like this. If they span, you get this light read, BioNano for comparison, and here the clipped reads for short reads. It is very good. It doesn't matter if you use sniffles or acute SV if you have high coverage. So these dots up here are 30 to 40x. You reach close to 100% precision and recall. If you have lower coverage, we would suggest to use sniffles, the red one, because it has sniffles, because it already performs with 5x quite good, while QtSV has problems with lower coverage. It drops quite a bit. At very high coverage, QtSV was a bit better, but we use sniffles because it performs well across a range of coverages. As you can see here, reaching 30 to 40x, you already reached the optimum of 92% precision and almost 92% recall. As a comparison, with short reads, you reach about 50%, but if at all, it's less than 50%. On a genome-wide level, you get about 10,000 SVs with short reads and 23,000 SVs with long reads per patient. That's a lot of new SVs. So for the last minutes, I have a few examples how that actually improves in the clinical diagnostics. The first two I will just mention here because I already showed you now examples for them. Compound heterozygotes can be phased now, so you do not need the trio to find out if they are on the same or on different alleles because you have haplotype phasing. That will reduce the cost for a lot of cases unless you are looking for de novo variants. Then you still need the trio. Structure variants and mobile elements, I just presented to you that we will have more than doubled the call of correct SVs. So a lot of new stuff to look at. I will not show a lot of examples here. I will focus on repeat expansions. They have improved a lot with long reads. DNA methylation and imprinting, and the so-called dark regions of the genome duplicate genes with a pseudogene duplicate. So repeat expansions, you can now with the long reads follow the following strategy. You can phase the two haplotypes, then you have potentially a shorter and a longer repeat haplotype if there's a heterozygous repeat. If it's a homozygous repeat expansion or no repeat expansions, both alleles will have the same length. You can generate the consensus repeat length for each haplotype. You need the minimum length causing the disease. Typically, that's published somewhere for the known repeat expansions that cause disease. And we currently use the tool Straggler for that, but unfortunately, Straggler doesn't give any graphical output, so we added that ourselves for our decision support system. So you see here the graphics. Most important, the right one. This whole thing was developed by Kaspar Thomas, now Chia, short Lani, and Leon also help with that. You can see the short and the long haplotype reads in a histogram. So the two alleles, the estimated size, and the size which is still wild type. So if you have 64 base pairs or one more repeat, you would be potentially affected by the disease. So here you see allele 1 is much longer and is a causal one. Looking at some cases, Huntington's disease, that was Tübingen case 15. We have a CAG repeat in the gene HTT. Here's the tree, the Stammbaum, the family tree from that, and the affected one that was sequenced. You see the two alleles in IGV, the expanded and the non-expanded allele. And if we look at our visualization of calling the causal variant, we see here, that's the one from the last slide. We have 152 base pairs in the expanded allele, 55 in the non-expanded, and it's clearly above the threshold of 63. So that case is solved. Same here, ataxia, spinocerebellar ataxia type 3, case 16. Here are the HPO terms. Here's much smaller family with a sequenced person. You see here the expanded allele CAG repeat in ataxin 3, 77 repeats. And here you see it in total base pairs. In that case, even the first allele is a bit longer. I'm not sure if that is enough to would actually cause the disease. The other allele is much longer, this lower one here. So, well, the second one is definitely expanded. The first one is slightly expanded. Next case, fairly new repeat expansions in the gene FGF14. So that one was published like 2 years ago as causal for ataxia. It's quite long, so you can see here that IGV has problems visualizing the whole thing. So it just shows you the insertion. Here 740, 60 something. And here's the other allele, which is a bit shorter. It shows you the lesion. Here's the long allele with a straggler analysis and visualization. It's very long, and straggler starts to have problems. It only found 5, or it only reported that 5 reads support the expansion. But if I count here, I see 10 reads. And you see that there are some mini-map alignment issues with the starting site. So we have to improve there a bit. We have to see how we can improve the tools to count all reads that support a repeat expansion. That even gets worse in the last example. Here, this is like a more than 10 kb long repeat expansion. And we only find 2 fully spanning reads. And then we see even in Nanopore a lot of clipped reads, which cannot fully span. And this is amyotrophic lateral sclerosis, ALS, in the C9orf72 gene. And you see that straggler completely fails. It calls 2 wild type haplotypes. So that's bad. We still have some room to improve, as you can see, for very long repeats. The easiest improvement will be to have longer nanopore reads, so to go to 25 kb. Another option would be that we look at the linked haplotype. Here's an example, which I will show a few more slides later. The expanded allele often comes together with a specifically linked haplotype. So you could actually use SNFs in the vicinity to identify a repeat expansion if it is too hard to identify it using this straggler algorithm. So you can see here in the many SNFs that that is a very specific haplotype here. Methylation is very easy. With the 5-base model, you get the methylation difference between a cytosine and a methylated cytosine. So we can then estimate the amount and use any sort of tool. IGV can plot that, but also other tools like MethylArtist. Caspar shows here an imprinting locus where you have 1 haplotype methylated and 1 not. So it's very beautiful for the clinicians. Here's an example case, autism spectrum disorder, the SNRPN gene. Here in that case, we have a combination of a deletion of 1 allele. And that means instead of having 1 allele methylated and 1 not in an imprinting region, you have suddenly everything hypo, so unmethylated. And that is explaining the disease, the combination deletion and the lost methylation. And here's another case, Fragile X syndrome. Here's again a combination of structure variants, or in that case, a repeat expansion, a CGG repeat expansion, which is homozygous because it's X chromosome male. And if you look at IGV in a control, you see a little bit of methylation. And if you look at the repeat expanded case, you see much more methylation. So in fact, the longer your repeat expansion, the more likely you get hypermethylation of the locus. So it's a double molecular phenotype, expansion plus hypermethylation. The mechanism is well known. That's nothing new. You can read about that. And there's even a difference between just the repeat expansion and then a long repeat expansion with the methylation. Then I mentioned duplicate genes. I will have only one example. We unfortunately didn't include much in the pilot, and we are now working on that in more detail. But one of the most famous genes with a pseudogene copy is SMN1 from spinal muscular atrophy. SMN1 is a copy which is a bit shorter, but you can have multiple of them, and it can even modulate, I think, the strength of the phenotype depending on how many copies you have. But for us in the past, it was just simply very hard to analyze these regions because all these SMN1 copies would align to SMN2 copies align to SMN1. So you could never be sure, for instance, if you had a clear-cut deletion. Thanks to the long reads, you can now uniquely align them to one or the other and anchor them in the surrounding regions, the long nanopore reads, and now you can clearly see deletions in SMN1 nicely, and that is the causal variant in that case. So last minute, I hope I'm not totally over time, but I have 3 more slides of our latest cool project where you will be able to read the full story next week in Nature Genetics. It was a peculiar case of a huge family that was collected by the group of Stephan in Utah. They have spinocerebellar ataxia type 4, and they are a large Mormon family which was originally coming from Sweden and now is in Utah, Georgia, and in other close by states in the US. And because the Mormons tend to have elaborate books on their ancestry, we have these huge family trees. So you would think this case has to be solved easily with so many affected people across 6 generations. But it wasn't. There was a 25-year or even 29 years since they first found this family, and it was not solved. So we started here with Olaf, Tobias, Nicola, to sequence that with PacBio, ONT, and everything. We already had at least a linkage region based huge family. And we discovered a new repeat expansion in the gene ZFHX3. If you look at all the affected people, they all have a GGC repeat expansion in one allele and none in the other. And these are the 2 unaffected family members. So all of them, based on Straggler and our in-house visualization, have this expanded allele, and it has to be at least 42 repeats, and healthy is 20. We could validate that by PCR amplification. Here, the unaffected again and the affected. We could look in our in-house database, and we identified more patients that had it based using Expansion Hunter on short reads. So once you told Expansion Hunter where it is, it actually could find them. And you can again see beyond 42, you have the disease. There was one case with 41. We don't know if it's a late onset person, but at the moment, the person didn't have it. It was there because of breast cancer. Then we looked at the haplotype. As I said before, you can use haplotype to better identify the causal variant. All the red dots are on the linked haplotype. All the blue ones are on the other haplotype. Here's the repeat expansion. And you can see that all these cases have a pretty long haplotype block, which still dates back to Sweden and Scandinavia, we found out later. There are some crossovers in some people, but you can still see that haplotype block. And you already see there's a case not from Utah, but from Lübeck. I mentioned that specifically because Malte Spielmann's group found independently in 2 people the same repeat expansion. We found that out during the study. We included them, and they have the same haplotype, indicating the same ancestor from Sweden somewhere. Now, looking in the database, we again could see, using also these linked haplotype SNPs, that there are more people in our database. Initially, we identified with Expansion Hunter 5 extra cases in 6,500 short read genomes. But then we looked in more detail in the haplotypes, and we found 6 SNPs here in the middle that are highly specific to that repeat. They are also very low in GNOMAD or 0, and they can specifically identify cases. So you could actually screen for the repeat expansion just using these 6 SNPs. And then we found 2 more that Expansion Hunter missed, but you can clearly see the expansion. And we are still screening for more cases. So all of them have that SNP. We also found that there is one SNP which seems to distinguish the German branch and the Lübeck and US branch. So there must have been one additional event in one person, but all of them, we think, based on these SNPs, have the same founder 200 years ago or so in Sweden. There's a bioRxiv paper out there, but as I said, I would wait till next week. The main paper will be released. So thanks to everyone who contributed. Lots of people from my institute, specifically Elena, who single-handedly made all the wet lab implementations. Now she got some extra people to support her, but initially she implemented all of that. Kaspar, Leon, Jakob, Mark, Vladislav, all of them are involved in developing the pipeline. So lots of bioinformaticians lately, joined by a few more people that are now working on repeat expansions and duplicate genes and so on. And Nicholas for organizing all of that in the NGS Competence Center. And by soon having our third Promethean, so there's a lot of work to do. And Tobias for organizing the longer consortium together with the people from the other university hospitals from Aachen, Charité, Berlin, and Hannover.